All right. Thanks, you guys. Hi, everybody. Thank you for coming to the 13th Southern Steelhead Coalition meeting. All right, so we have some really fun things to talk about today. We'll have a quick hello and updates as we wasn't a lot of updates today, but we have some other things to update you about the Southern Steelhead Coalition. We'll talk about the Southern Steelhead Symposium, which is our big update. Then we'll go into our speaker, Brooke Penaluna, who is a research fish biologist and lead scientist for the J. Andrews Experimental Forest and for the U.S. Forest Service. Her talk is going to be on what's really in the water, testing eDNA for stream biodiversity. Then we'll have a question session for Brooke's presentation discussion, about 20 minutes. We'll have a small strategic plan update. And then this will be the time of coalition-wide project updates. We would like to hear from agencies, governments, nonprofits, other organizations. And then finally, at the end, will be open discussion. This is when, if you have an announcement, if you have a topic that you would like to bring to everybody's attention, please, this will be the time for you to do so. So let's first start with the Southern Steelhead Symposium. So last year, we had a Southern Steelhead Symposium on September 30th. About over 80 people came. A lot of you that are here on this call were present, so I want to say thank you to that. This year, if you didn't get my email, it will be on September 15th, the same location, about roughly the same time, a little bit longer, be 9:30 to 5 o'clock. And this is going to be steelhead centric, and I mean Southern steelhead centric. So if you have a talk, we're looking for speakers. We have a few speakers lined up, but we are needing more speakers. We're looking for speakers, tablers, and poster presenters. If you have a Southern steelhead poster that you've been wanting to give, or you want to make a poster for this is your time now to do it. And this is going to be all about posters. It's all about steelhead. Now, when you give it, I wanted to do a caveat for talks. We're making it very steelhead centric, right? Steelhead are the star of the show. So if you're doing a restoration project, it's about steelhead mainly, but also about other things because steelhead do not live in a vacuum, right? They're ecological being, right? They affect so many other animals. So if you're like talking about how your restoration project is saving steelhead and condors, that's fine. But we want it to be super steelhead centric, southern steelhead centric, not anything about the northern population of steelhead. We want it to be based on southern steelhead work. Now, if you want a table or have a poster and you're not super steelhead centric, but it's work that involves steelhead, that's the place you should probably angle for is tabling and poster presenting. The presentations are going to be a lot more open if it's about restoration work in Ventura and you're removing invasive plants and that helps steelhead. That's a fine poster to give. If you're doing tabling about work you're going to do that you do in Ventura County or you do in Santa Barbara, that's fine. We're a little bit more open to that, but for the talks, these talks steelhead has to be the star of the show, or the headlining act, right? So if you're wondering if you should give a talk, yes, please give a talk. We have a few more spaces open, and this is going to be a long day. We already have quite a bit of people sign up for registration, which is great. And it's still a couple months away. So if you haven't had a chance to sign up or look in the forms, this is your time to do it now. I sent out on the agenda all of the links for registration, for volunteering, or not volunteering, sorry, registration, for signing up to speak, to do applications. So we're really looking for registrations of you coming to it and also people signing up to be a speaker. I will take questions at the end of this if anyone has questions about this, but right now I also wanted to do a call for taco recommendations. We're going to have a taco bar. I'm looking for places to cater. If you know of a great taco caterer, so we can have a great taco bar, reach out to me. Email me. You guys have my email, but we're looking at that. Does anybody have any questions about the Southern Steelhead Symposium and what talks or anything like that, or what a talk entails, or anything to that nature? Okay. All right. If you have a question, you can email me about it, but please sign up. I will also show you how to do that. So give me 1 second. Ba-ba-ba. Okay. So hopefully you all can see this. This is the Southern Steelhead Coalition website. And when you come here, southernsteelhead.org, you go to the About section and you go for Southern Steelhead Symposium, okay? So and when you click on that, not only does it give you a little page about Southern Steelhead, it gives you the information and archives of the first one. And then you can go to the second one, and the second one will have all the registration links, time, flyer, where, when, where. So this is where you can RSVP or register, and this is where you can sign up for presenting or tabling. So if you want to be a poster presenter, want to be a speaker, that's where you can do it from. All right. Any more questions? Any more questions, anybody? Okay, great. And anybody who came in late, please put your name and affiliation in the chat for me so we can make sure that we have a better head count of who's coming to these meetings. Thank you very much. All right. We move on from that. Okay. See chat. Okay, great. Other than that, hope to see you in September. It's gonna be a great time. We're looking forward to it. We already have some really great talks. We just need to fill out the rest. So please register for the symposium as soon as possible and let us know who's coming. We really do need to know who's coming for the taco bar so we can make sure that we're feeding everyone. Okay? All right. Let's move on. So again, the Southern Steelhead Symposium, September 15th. Register the attendance speaker and sign up to be a speaker. We'll be talking about, after the talk, we'll be talking about the talk given by Brooke. We'll be talking about about coalition involvement and expansion. What meetings do you think we should join? Any outreach events that the coalition should be a part of? Let me know. And then finally, our next meeting date will be the symposium. And then after that, our next online coalition meeting will be in November. And I will send out an invitation for that. Again, if you think somebody should be on this meeting, feel free to send this meeting invite to all interested parties. Tell them you can even email me to have somebody sign up to be at this meeting. You give me their email and we'll put them on the email list. Okay, with that, I would like to now welcome our speaker, Brooke Penaluna. And she will have the floor. Thank you, Brooke. Oh, I'm sorry. Again, forgive me. I stopped sharing too quickly. I want to introduce you. So again, she's a research fish biologist and lead scientist for H.J. Andrews Experimental Forest Service. And her talk will be on what's really in the water, testing eDNA for extreme biodiversity. Thank you, Brooke. And feel free to start sharing when you need to.
Sounds good. Thank you, Cam. Okay, perfect. We're there.
Yeah.
Thanks for the introduction and the invitation to come and talk with you all today. I think about what I'm going to talk about today as kind of like a primer and what is eDNA, what's it good for, what can it do, and then I'm hoping we can have conversation or questions at the end about, how you might want to apply this in your area of research or your area of monitoring to get at southern steelhead. So can talk more about that. So in general, globally, we are seeing a decline in biodiversity across marine, terrestrial, and freshwater ecosystems. Among them, our freshwater biodiversity is the most imperiled. And that's likely because of the high level of endemic species that we have in our freshwater environments, as well as our cryptic species. Traditionally, when we sample in the field, our traditional methods are based on visual detections and counting, which isn't always standardized, and it's dependent on that practical and taxonomic expertise of those that are in the field. So when you catch a fish, you need to have the person in the field, being able to identify the different features that make it, identified as one versus another. They're often limited to assessments of adults in the summer and often of species that are listed at elevated conservation status. And I should note, in the net on the left of the electrofisher is a steelhead from the Willamette River. So what is eDNA? It is short for environmental DNA. It's characterized by a mixture of genomic DNA from different organisms. And so it's DNA that can be extracted from water, soil, or air. And water contains animals, plants, microorganisms. And most of the eDNA that's in the water lasts for 3 to 60 days based on a variety of research that's now come out on this topic. The first paper that was actually published related to eDNA came out in 2007. So now we're coming up on about 20 years of this technology being out. And initially when it came out, it was still sort of being sorted out about what it can and can't do. But now that it's been around almost 20 years, we have a really good sense of, its strengths and weaknesses. So my talk is organized to explain what eDNA can do, and I'm going to show you 2 examples at the end after I kind of talk through what it can and can't do. So before I kind of dig into the examples I'm going to show from streams and rivers and lakes, I wanted to just say that eDNA is being used, in ports. They're sucking air out of, shipping containers and trying to identify exotic species arriving on shipping containers. It's being used to, they take snow and they'll melt snow and they're identifying which paws that it is to identify, big cats that roam across places where you don't actually see them because they're quite elusive. And so that allows them to get at the information about what species that they actually are. So that's just an example of how it's being used elsewhere. So what is eDNA? The most commonly used method for eDNA is eDNA barcoding or single species eDNA. Those are the same thing. You collect the water in the field. You can collect it in a water bottle and filter it through your filters, or there's direct filtering systems in the stream. You extract the eDNA back in the lab. And then with the single species approach, you essentially look for a fluorescence on the machine of that single amplified gene that's going to be quantified. And so in this type of work, you do all the work in advance in the lab to create a primer that's going to identify the specific species you're interested in. So for example, if you were interested in steelhead or rainbow trout, you would you look for the genetic information of them and you would distinguish it from related species. In my system, I have to, for example, separate it from coastal cutthroat trout. Those don't swim in your system, but essentially you're trying to make sure you don't get detections of similarly related species in the development of the primer. You don't want sister species showing up. So the example that I'm going to show here is an example of coastal cutthroat trout. They are the trout that swims the highest up in our stream networks. And so we did a study comparing electrofishing to eDNA for detection of them, trying to understand their distribution at their upper extent in the watersheds. So this is what our sampling approach looked like. We sampled along a stream that you see there down the center of the stream. And with electrofishing, it was continuous sampling. And we counted up how many fishes we detected and what species they were between the different points that were 50 meters apart. So each of the numbers on here were 50 meters apart. And so you'll see our electrofishing results show a dark blue bubble in the center, and that means it was coastal cutthroat trout. And then we had this halo because we can't always distinguish young of the year of rainbow or steelhead from coastal cutthroat trout in the field when they're young. So the halo is young of the year. It could be either of those species. On the eDNA side, we're able to get at that genetic distinction between the two species. And so on the left side, it's presentation of only coastal cutthroat trout results or detections, and you're able to see that we detected coastal cutthroat trout at 4 of the sites in this example I'm showing here, with electrofishing, we detected it at 3 of the 3 sites. And then above that, where you're seeing the gray dots, it was no detections with either method. So we did this in 60 streams throughout Western Washington and Oregon, and essentially trying to understand, the comparison of the 2 different approaches and how well eDNA worked at the uppermost extent of fish. And we found that trout eDNA was detected above electrofishing uppermost fish in over half of the streams. And that was by 50 to 250 meters. And so you're able to see some examples of what that looks like here. So you're looking at results kind of shown as a mirror. So if you first look at the electrofishing results in blue on the right, you're able to see where the blue dots are detections of coastal cutthroat trout, or the young of the year is shown in the halo again, and gray dots are showing where we didn't detect. So where we have blue, you're seeing the detections compared to the eDNA on the left side, and you're able to see detections and then different level of detections. So essentially in all of the replicates that we took, since we wanted to understand how this compared and potentially how many replicates you needed to take in the field, to be able to be sure that they were there, you see that the way that we're presenting the data is showing detections in one replicate versus up to 9 different replicates that we're showing here. So another eDNA method that's used in the lab. So I should say there are just there's 3 main methods, 2 of them that I'm presenting today. Those are the main 2 that are used. So there's that single species I just presented, and then there's what's called eDNA metabarcoding. And with the first method I presented, I talked about how you have to have all that work up front, developing the primer to focus on the one species you're interested in. And here with the eDNA metabarcoding approach, you do something different. So the difference here is in the lab, you collect the samples the same in the field. But what you essentially do in the lab here is you actually want to find a place where you can distinguish similarly related species. So in this case, my example is coho with coastal cutthroat trout. And you would look for related species, on one gene. And so this is just like the 12S, and that you're able to distinguish multiple species from the one primer that you're developed. And I want to sort of talk about like the next level that's happening in the research world, which is taking eDNA metabarcoding and adding it up so you have more. So you end up with what are called multi-gene eDNA metabarcoding. So essentially you're looking for your species, but on multiple genes. And the reason to do that broad technique is so that you can get at you have this built-in redundancy, and you can get at more information because you're using multiple primers. So in this example here, on the left side, I presented the electrofishing data in the field, and you're able to see that all 3 methods in this one stream detected coho, coastal cutthroat trout, and our rainbow trout. All 3 detected that species. And then if you look on the right, what you're seeing there is the approach we use that is called multi-gene eDNA metabarcoding. And you're able to see that if somebody were to say to me, how sure are you that you got a coastal cutthroat trout in my stream? I can say very sure because I detected it off 8 different primers and in so many replicates that I took. And that just becomes important, especially as you're dealing with, endangered species, threatened species, or species that are more cryptic. You want to have more assurance that what you're actually detecting from the water sample really is something that's there. So that's why we've started to do this multi-gene eDNA metabarcoding approach. So going forward in the talk, I'm going to present results using the metabarcoding approach. So what we show here is that using this approach, eDNA can distinguish species across taxa. And so you're able to see essentially from one water sample, we can detect fish, amphibians, crayfishes, mammals, pathogens, and across the different streams. And so what you're looking at here is electrofishing results on the left side, eDNA results on the right side, and each of the different columns is a different stream. And so you're seeing the different columns from the top to the bottom, and you're able to sort of compare the detections about whether we detected it with electrofishing and with eDNA, and essentially which species. And so what we see is that eDNA actually increases our taxonomic resolution of what we're able to detect in the field. And so you'll notice that coastal cutthroat trout, which is the second row, has detections across all four for both electrofishing and eDNA. And we're getting detections for rainbow trout or steelhead, our omicus detections, in two streams that we didn't necessarily detect it in the field. We had what we were calling YOY, or the young of the year, but we weren't able to say with assurance whether it was coastal cutthroat trout or omicus. And with eDNA, we're able to do that. So we get that extra level of taxonomic resolution. We're also able to detect sparsely distributed species. The example I'm showing here is mountain beaver, but there's a variety of other examples, of different animals that you might look for that are more rare or sparsely distributed that you're able to detect and get additional information. It can also help you understand something about cryptic lineages. So on the left-hand side with our electrofishing data, when we sampled in the field at these four different streams, we brought the sculpin bodies back for Doug Markle, who of the Oregon State University ichthyologist lab. He's since retired, but he identified from the sculpin bodies torrent sculpin that he said was in one of our streams, and in the other stream he said we had reticulate sculpin. So we had those two species identified in the field, or so we thought. And with our eDNA results, what we're finding is we don't actually even have torrent sculpins in our stream. Genetically, that's not what they are. We do have reticulate sculpin. They're only in two, but we also have riffle sculpin in three, coast range in two, a prickly-like sculpin in three, a margin-like sculpin in three, and then this sculpin spa ends up being 4 to 14 additional lineages that I've just classified into one because they came mapped from all over places in the world that didn't line up with where they should be in the Pacific Northwest, which in a lot of ways just says there's some, undefined resolution related to the sculpins, the sculpin taxonomy, and that they're probably likely differentiating now in the field. But the detection of the torrent sculpin not being found was actually reinforced by work that Michael Young did out of the Rocky Mountain Research Station, which he said that torrent sculpins are actually only up in Washington, and what we have in Oregon are actually a different species, and our eDNA work is supporting what was found with that other population genetics work. And another thing that's really great that you can do with eDNA metabarcoding results is you can get at a sense of genetic diversity. And so what we did here was with some of our samples that you just saw before, we're able to get a sense of the haplotype diversity, which is just another metric, for understanding our species. And there are, various reasons why eDNA haplotype diversity could be useful. And it's the type of diversity you can differentiate with eDNA, because right now eDNA is based on the mitochondrial DNA. So it's easier to sample than having to capture animals and get a clip or snip donation from them. You don't need permits or permission, to sample like you do if you were going to do some other population genetics work with the fish. But what we show here is that just for this example, we also did this for coho and chinook and rainbow and steelhead in the same paper. So if you're interested in seeing some of the diversity of the other fishes, we found that some rivers have what are called private haplotypes, which they're sort of unique genetic signatures. And so we had one unique private haplotype in a coastal river and one in the Umpqua River. And this kind of tells us something about which rivers might have historically shared genetic information, or where our fish is sort of moving for. So we're able to see, moderate diversity in the Willamette River and the Umpqua, but it's not shared, so that says something about how the fishes are, mixing or not mixing. And then we saw the lowest diversity in the Rogue River, but they shared some sequences with coastal streams or the Willamette River. So those fishes had either historically mixed to share that genetic information or are currently. And so I said in the beginning that I was going to talk a little bit about eDNA and kind of what it could do, what it is good at. And then I was going to give two examples to kind of show, like, I just think of it as like next level where you're answering kind of applied questions where you're trying to use it as a monitoring approach or technique to better understand something. So we asked the question related to habitat diversity. Essentially, more diverse of habitats that you have, does that mean you have more biological diversity? And this is a big question that's being asked with river restoration. And so this is the Mackenzie River and the South Fork Mackenzie River here that flows into the Willamette River. And you're looking at pre-restoration water with the blue color. So it was a single thread channel that flowed directly into the Mackenzie. And post-restoration, you're going to see the flow paths there where all the purple is kind of showing where the water was after the stage 0 restoration site went in. So we measured pre-restoration, so prior to the floodplain river restoration, and you're able to get a sense of what species were found in the South Fork Mackenzie. And you're seeing the biggest diversity of our fishes down near the confluence, which is what, you know, any of us that work in rivers and work with fishes expects to see. You always have higher diversity in places like that, and that's what we're seeing here. For your guys' interest, we have steelhead that are found, you know, all the way up at all three of the sites that we sampled prior to restoration. And after restoration, we actually see more species and taxa present than we did prior to restoration. So just as a reminder, this is what it looked like prior. And this is what it looks like post. So we end up with a greater distribution of those species and additional species and taxon that weren't detected prior to restoration. And so we're getting more species and more detections. It should be noted that post-restoration, we also have non-native fishes, you know, that are able to access these waters just like our native fishes. And so we had addition of those species move into these systems. So the last example I wanted to talk about was the relationship between freshwater biodiversity and watershed stand age. So there's been a lot of work, you know, in terrestrial ecology and forested ecosystems that are relating terrestrial biodiversity to the age and structure of forests. And so I wanted to do the same thing relating freshwater biodiversity to watershed stand age. So we had some different hypotheses developed in advance about what we might see for this work. So you're able to see the streams that we used for this assessment from coastal Oregon, and the size of the bubble, the smaller the bubble indicating that site, was a younger aged forest all the way up to, you know, forests that were just about 125 years. And so we're essentially looking at the relationship of biodiversity to watershed stand age. And so we had some hypotheses about how it might, they might be higher with older forests, or they might be higher with younger forests, or maybe the, you know, mid-age forests, because we end up with both the pioneering species and the more specialized species. Or maybe there's no relationship with forests because it's, you know, streams and the relationship's not there. And so we had this watershed mosaic. So it's more related to other things that have to do with the watershed than it actually did with forest stand age. So before I show you the next slide and tell you what we found, I want you to think about, you know, what you actually think the relationship might be, which you think it's going to be higher in. We found higher richness for both invertebrates and vertebrates in watersheds with older forests. So what you are looking at here is taxonomic richness on the top panel, and you're looking at functional richness on the bottom panel, and you're able to see for both invertebrates shown on the left and then vertebrates on the right that it's higher in older forests, which is really, really neat and actually supports a lot of conceptual ideas that people had, you know, for the last 20, 30 years, but hadn't actually been shown in their literature, is how tightly coupled likely our freshwater environments actually are with our forests. And that in order to have higher, you know, biodiversity, watershed standage is a part of that equation. And so just kind of taking all of these pieces together, you're able to see that we can actually, get at and understand more about freshwater biodiversity by using the multiple primer sets from a single sample. You're able to see that from the work I presented. I do want to say, though, if you guys with the different work you might be doing are interested only in one species, the single species approach, would be the way to go with what you would be doing. I showed that there's this world of like hidden biodiversity in freshwaters, and that, eDNA can allow us to understand more about cryptic species complexes, distribution extensions, detection of rare, sparsely distributed, and overlooked species. I should just add that it is really good at the distribution piece. So if a lot of your questions that you have are, where are southern steelhead in your different systems, eDNA might be a good approach for you to do, because it does really well at getting at distributions, especially for fishes when they're in like low numbers, 1 and 2 fishes, and you're able to detect them in the system. And that eDNA information can allow you to understand a broad assessment of freshwater biodiversity, and answer additional questions that one might have, so you could prioritize different taxa on different watersheds or on our landscape. And with that, I'll take any questions.
Thank you again. Thank you again. Now this is time for questions. So if you guys have any questions, we have a lengthy question period, so you can get all the nitty-gritty out from Brooke. Tevin, please.
Morning. Thank you so much, Brooke. Sorry, going through a cold right now. My voice is screwed. Um, you mentioned identifying sparsely populated species during the study. How many samples did you take for species like mountain beaver or giant salamander?
Yeah.
And did you collect in multiple locations for these species to get an eDNA detection?
Yeah, it's an excellent question. So in the study that we've done, and in a couple different studies, we found that 3 replicates tends to be the right number for detection of the species when they're in low densities or low numbers. If you go above that, that's only going to increase your resolution. I mean, just think about, it's like, like with your electrofisher, the more passes you make, or the longer you actually electrofish at the place, you're more likely to find, the fishes that are there. So it's the same idea, but 3 seems to be a minimum number, with increased past that, just improving that. But I would say 3 at a minimum. Some people still go out and might only collect 1 replicate, like to try to understand something with spatial resolution. And you might miss species with that. You're missing some detections when you're doing below 3. So that's what yeah. Did I
Thank
get
you.
at your question, Tevin? Yeah.
Yeah, the follow-up was, did you sample in multiple locations for those
Mm,
species, or were you able to pick them
yeah.
up from one
Yeah.
location?
So we do sample, along streams, along rivers, in large water bodies, reservoirs, lakes, and some of it has to do with the geomorphology of that different stream system, and like the flows at the time of the year. We find that when flows are higher, so think, winter, spring flows, when flows are higher, signals traveling further. When it's, summer, fall, before the rains come in, you're in seasonal low flow. The signal's not traveling as far, it's like less than 50 meters, we're finding during that low flow, the signal's traveling, where sometimes it can be up to a few kilometers at higher flow. In rivers, we found signal up to 25 kilometers away. We took fishes we didn't have in our system, put them in cages, and tried to detect, how far we're detecting the signal. And so it depends a little bit on your system. And I think what your question is, if you just want to have a sense of if they're there, is the question, or sampling just above a confluence enough, or do you need, do they use different parts of the stream network, and it might make more sense to increase your sampling along the stream?
Yeah, I mean, this is a very specific project. I'm kind of in the I guess study planning phase. We're going to do a flow corrected eDNA study on smolt out migration for steelhead in the Santa Paula Creek watershed. But there's kind of a question for the U.S. Fish and Wildlife Service if a certain species of sucker is in that tributary. Where they haven't been able to identify between the two that are in that genus, and figured I might as well grab some samples while I'm grabbing, eDNA samples for steelhead. So thank you very much.
Mm-hmm. You might even be able to use the same sample if you have enough DNA, or if you just grab enough replicates, you could, have enough DNA
Nice.
from the lab. Yeah.
Thank you.
Yeah.
Awesome. Okay, next we have Mark Rockwell on the list.
Hi, thank you, Brooke, for all this great information. And I was particularly struck by the end of the discussion having to do with the age of watersheds and the diversity within those watersheds and the role that age plays in diversity. And I just am curious because we're in this, what I would call the federal um, push to utilize public lands and forests more extraction methods. And how do you offset how the Forest Service works in that situation, knowing now that increased age of the forest creates much more diversity and health, watershed health?
Yeah. I mean, what you ask is a really on topic question for our moment. And I think we can inform managers that, leaving our older forests does actually increase this diversity. Name the species that are found in that diversity mix and the watersheds of interest to them, which, gives them justification to not harvest, and potentially push back against, harvesting everywhere. It might help us be able to prioritize certain locations or forests that are older. So I think that's probably the best way
A
to use
tricky,
information.
That sounds
Yeah.
reasonable.
yeah,
Mhm.
a tricky answer.
Policy people have to follow
Yeah.
the directions, though.
It's a tricky question, right? It's hard. Management versus, management, that's the internal, that's the age-all thing, is management and making sure we follow the rules, right? Next is Rosie.
Good morning, everyone. Brooke, thanks for that presentation. Really interesting. I want to go back to what you were saying about distances and correlating that to flow. So, do you have a range of CFS that you use as your criteria to help you determine whether you sample every 50 meters, every 100 meters, every 200, or whatever?
I don't. I just normally base it on, I feel like we should, as a research community, we should work towards that because that would help managers be able to kind of select and pop it in. It's a good idea and actually makes me think, do I have enough data that I could, get at something like that to say something like that? So I like the question because it helps me think about like something that I could do next, you know, that would be useful. Offhand, I don't.
Because I think
Other than, yeah.
I think my concern is that, a lot, as we're sort of setting up these study designs and sampling protocols, having a better understanding of how much we can have confidence in our data or not is really helpful. And it's the one hiccup with eDNA that I think has plagued us ever since this first started. And I'm just wondering if anyone knows of any sort of reliable research that kind of addresses that question of detectability and distance sampled.
Well, I mean, I think different research projects address the detection, the ability to detect with their different method within each of the studies. I feel like that's kind of how most studies are now done in the last, 3-5 years, solidly identify how well that they can detect. Because as a research community, we sort of learned we weren't doing very well. I want to say like 10, 15 years ago, in that wasn't being properly addressed. And so I feel like every study has that detection piece set up. But it's less understood, across systems. Can you essentially generalize to CFS levels and different geomorphologies and say something about how you should do it in the different systems? And I think right now we don't have that. And I think the best that you could do is just have a sense of, I'm going out when the flows are higher. And so I need to sample less frequently in spatial resolution. But the question would be, is that 2 kilometers in your stream, or is that
Right,
5?
250 meters. Thank you.
Yeah.
Thank you.
Great. Tim Robinson.
Good morning, everybody. Thank you, Brooke, for that very informative talk. Very appreciated, considering that all of us are struggling with eDNA and trying to figure out how to apply it in our own system. So I have a couple of quick questions. One, when you were talking about at the beginning using electrofishing and eDNA in parallel to see how well you're doing, are people using eDNA for calibration sampling with snorkel surveys? Now, eDNA, of course, doesn't give you numbers, but it tells you presence absence. So what it occurred to me is that maybe in some of our snorkel surveys, we should be taking eDNA samples simultaneously to see if we miss something, we may pick it up. And I'm wondering if you know of groups that are using eDNA in a calibration format or other sampling methods.
Yeah, I've seen eDNA compared to boat electrofishing, backpack electrofishing, minnow trapping. I don't know if I've yet seen it compared to snorkeling. I know people will often sample while snorkeling, but I don't know if I've ever seen the results come out directly comparing it. I think the thing you would have to be concerned about is that the snorkelers aren't dragging signal on their suits. Like you would have to sample with, you'd have to take your eDNA samples before essentially the snorkelers would arrive, so that as they're moving in the stream, they're not dragging the signal further up, and then you detect, above where maybe it would be seen. So I think you just have to be strategic about when you sampled with eDNA. Maybe there's one person who runs, ahead, gathers samples at set distances, and then runs back down, and you start your snorkel pair up the stream type thing.
I can see that as being very beneficial, so
Yeah.
I'm thinking about it. Follow-up question is on the multi-species primers. Can you give us a little bit of a cost estimate for those of us that are thinking about it? What are we looking at? How heavy is the financial side of this?
Yeah, I think if you're starting, the first thing you want to do is identify the lab where you're going to process your eDNA samples. And you need to figure out how the eDNA sample needs to arrive at their lab, because everybody kind of has a different vial size that they want to use, or a filter pore size, or how it's handled. So you want to identify that first. And then you want to look in the literature or talk with the lab, and since they're going to be processing the samples, and make sure that the meta barcode is already published in the literature, and have a sense of what it's able to detect. There's a lot out there now. I mean, since this has kind of been a technology that's been around now for a couple decades, there are a lot of meta barcode. For example, there's one that does like all ray fin bony fishes. So every ray fin bony fish, it has a sense of detection. Sometimes if it doesn't detect well to the species level, it will detect to the genus level. And at least you have a sense that, those species, or I'm sorry, the individuals of those genus were in there, if it isn't able to detect to the species level for some. And sometimes the reason it can't do that is there's just not enough, and I didn't talk about this at all, but the libraries, when you do the meta barcoding approach, the big work is actually on the bioinformatics end. You essentially get all these sequences back, and then you have to process it, through some programs and to identify which species are there. The benefit of having done that is you've sort of you have a data archive of every species that was there, that you could look for with that barcode. So I just say that because sometimes people will say, well, were there smallmouth bass there 10 years ago? If you had met a barcode, you could actually go back, in time and actually look at, because you have the data archived, you'd be able to look at that. Or were New Zealand mud snails there? If you looked for it in a barcode, you can get that information back in time. So that is a positive of it. But that library piece, sometimes we need more genetic information about some of the species that are just less studied, or there's less information known, like our sculpins up here right now. We're kind of, trying to figure out our sculpins. And so we need more sculpins that come in from the field that are identified by a taxonomist, linked to the genetic information. So that's one thing that we're working on a lot in the state of Oregon, is that every species that we can identify in our state, we want, to have genetic information associated with it to really be able to understand the information we're getting out of DNA. I would think at the, California level, you guys you have a lot of species that overlap with us, but you're going to end up with different genetic, diversity or signatures even within those species. And you're going to want the same thing done at your state level if efforts aren't already there to do it. So.
Excellent. So that sounds like that labs are all getting the same amount of library for the coding that's necessary to do detection. Because that beforehand it wasn't that way. Certain labs had more things than other labs.
Yeah.
So, but it sounds like we're moving towards everything's publicly available, so you don't have to search around to find the lab that's got the most capability.
Well, I think in some ways you still have to do that, because some labs specialize in one or the other. Some do both. Some only do single species and some do metabarcoding. Some don't always do both. But then you have so I think you just have to ask around to see, who's doing what, and having a few examples of the work coming out of their labs. And that would be enough to say, okay, that, they're producing the kind of things that,
I'm
give the results that I am interested in. Yeah.
Yeah. Great, Brooke. Thank you. Complicated discussion. Appreciate it.
All right. Thank you so much. This is really good. Russell, and then I have one question for you.
Morning, Brooke. Thanks for joining us today. And something that Tevin suggested, and I wonder if you come across it or considering it, or even in the process of maybe trying it, is single point high frequency DNA sample collection with paired flow measurement, which allows you to then begin to add a spatial component versus just presence. And I was actively searching through my files because there was one study done in Alaska that specifically did this, and it moved away from. Yeah, you seem to be great. So I'd love to hear if you've thought about that or any.
Yeah, I think you're talking about it was on salmon out of the Tal Levy's lab. I think Levy was the first author of that paper.
Yeah.
That essentially talked about the importance of detecting eDNA with different flows. They were measuring through multiple seasons. I think there's a few things just to note, especially as you guys will be in systems measuring smolts, but dead animals give off tons of DNA. So that's an important thing to note, because if you have adult spawned out fish in the system near where you're measuring for your smolts, you potentially are just going to blow out you're going to get so much signal. And Tal's, work actually showed that. He talked about the spikes you could see essentially from spawned out salmon, and then trying to detect juveniles and smolts and different things in the different systems. So I say that, and then he found that correcting by flow gave them better detection levels, just because they were measuring over a longer time period and not confined within a season. So I think if you have a study that's confined within a season where the flows aren't that variable, might not be as important to correct for flow, because essentially you've corrected it by the scope of your study. But if you are going over flows, or in where you might have spikes of rains and that kind of thing in winters, I think it's probably something to look into and would be important. But also just think about that spawned out salmon piece and where those fish would be in context of your samples.
Yeah, I appreciate that. That's a really good insight. I mean, our systems are super flashy anyway. So within any given season, we're seeing like, low base flows and then spikes. And so correcting for flow is kind of adjusting that. Have limited adults at best, maybe one or two showing up in a system, and then, few smolts as well. And so really trying to to add a little bit extra dimension to it to say, instead of just like, we know Omicus is here, beginning to see if we can't, make them more descriptive. So I appreciate that. That's really good thoughts.
And the other thing to note is it also detects eggs in the ground. There have been some studies in Washington that they used it to identify reds. So I'm not sure, just in what you would be doing, if that's something to think about as well.
No, that's awesome. That's an interesting thought. I was at ESA this past year and was at a talk where people were doing bird observations with eDNA strips just around a prairie and doing bird detections that way. So it's such a powerful and growing field. It's cool that you joined us. Thanks.
That's amazing. Brooke, I have one more. Oh, I have a question from me, but I don't know PSMFC is.
This is Mark Capelli.
Oh, hi, Mark. Okay, yeah. Go ahead. What's your question?
Hi, Brooke. Thanks for that presentation. It was quite interesting. I've been following this eDNA stuff for a while, and it's really advanced, it seems like, in the last 20 years. You mentioned in one of your slides detecting specific haplotypes, and we have an interest down here in Southern California, Steelhead, with different haplotypes associated with resident and anonymous life history forms. So I wonder if you could say a little bit more about the primers you're using for those haplotypes that you were looking at.
Yeah. So eDNA right now has been based on the mitochondrial DNA, because that cell wall that comes, the wall that's around the mitochondria, tends to persist longer in different habitats. It doesn't break down quite as quickly. So that's why it's really useful to use the mitochondrial DNA for eDNA. I say that because in the last 1-2 years, people are trying to do nuclear DNA to get at diversity, which would tell you more about, like within population diversity. When you're using mitochondrial diversity, that allows you to estimate the haplotype genetic diversity that you and I have just been talking about. So I just wanted to sort of say that first. So for us using the mitochondrial, we found that ND2 was the best at showing interspecific diversity in Pacific salmon and trout. So we actually had Omicus, coastal cutthroat, Chinook, and coho, and that was the best one. COI or 12S contained differences among species. So they did the best at species presence or absence, or did adequate to tell you if it was there or not, but didn't give as much of that diversity piece. We couldn't tell as much about haplotypes from those other genes. I think potentially there might be, that paper was now 5 years old. So there might be some other work out there showing other genes, but I would start with, ND2 as you start thinking about that. And one other thing to note that and you might be aware of this already, is right now using the mitochondrial DNA, you can't distinguish between whether it goes to the ocean or not. Essentially an Omicus is an Omicus. You can't tell if it's a resident, a river, a sea run. It's just an Omicus.
Yes, I think that is good. Mark, if you'd like to follow up on that. Okay. I think we'll get back to Mark if Mark has more questions. Finally, Brooke, since you are very seasoned in using eDNA, if you could give us the must-haves for an eDNA collection kit and the things to stay away from, in your opinion. Like, what's the easiest, what have you found that just does not work when you're out in the field, and things that really do are like, I really needed this hand towel because XYZ or something like that.
Well, I think the most important thing is that you identify the lab that's going to process your samples before you go in the field, and that you collect according to their protocol, because every lab has subtle differences. And you don't want to come back in, end up with samples, and then try to go with a lab that doesn't want the samples coming in that way. I say that because, and I don't think that there is necessarily a best. Some people like to put it in, like their samples to come in from the field in alcohol or in buffer or in desiccant. In reality, it's all working, like all that, it's shown that it all works in the different settings. So that's why I would say that's the most important piece. If you store it in alcohol, which is a common way to store it on gravel roads, there's a chemical reaction. Essentially, you're creating a centrifuge in the back of your truck. And we found that out the hard way, that you can actually, centrifuge your alcohol stored samples. So we don't store ours that way anymore. So that's the only thing I would say, because there are a few labs that are adamant you use alcohol, but I don't think they're sampling on a lot of gravel roads.
Yeah, that's really good to know that you've lost a whole sampling day because of physics.
Yeah.
Okay.
Yeah.
Mark.
And that has to do with the sample,
Mhm.
the filter chemistry interacting with the alcohol, the bumpy.
Great. Honestly, lessons learned like that, little things are really important. Mark, I think that you're unmuted. Is there anything follow-up you have to that last question before we move on?
Yeah, I was going to ask if there's any interest or plans to um, deal with nuclear DNA using eDNA techniques that would allow us to get at particular haplotypes and in particular an inversion that has been very instrumental in figuring out why fish go to sea and don't. And if that is an area that others are right interested in and trying to apply eDNA techniques to.
Yeah. So right now with eDNA samples that are being collected from the mitochondria, you can get at haplotype genetic diversity. You can do that now. And if you did that, I would suggest the ND2 gene, assuming that you're trying to get at Pacific salmon or trout. For the nuclear piece, that's where you're going to be able to tell more about which ones, the life form essentially of the species. And the real issue is that the nuclear DNA doesn't stay around very long and it's broken down in the environment. And so it's kind of it's one of those things that I think it's trying to be pursued a little bit, and at the same time it's elusive to actually get nuclear information in from the field that hasn't broken down on time to be able to sample. So I think it's something that's being progressed, and I think I am not sure, I know that it's a conversation piece that a lot of people who do eDNA research are having. Like, I'd love to pursue it a little more, but right now I can't get any of that work funded. So we just try to do it when we have a few extra pennies, from things. We'll try to test for it and see, just to kind of advance it a little bit. But
what generally is the breakdown rate for that nuclear DNA?
Well, I think the issue is that it still has to be developed. I think that's the real issue with nuclear, eDNA is essentially because you're trying to detect nuclear signal in the field that's just not as present as the mitochondrial DNA is. So I think that's one of the first issues. And then if you're able to detect it, it follows on, population genetics approaches that are, well vetted. And so I think the techniques are there for what you would do in the lab. It's just potentially getting it from the field or figuring out if there's certain habitats that might, I don't know, have more nuclear DNA buildup because they're breaking down less quickly than other parts of the environment. Like, I think it would be worth testing in hatcheries, right? In places where you just have a lot of DNA, you'd have nuclear DNA. That would be the place you'd want to start practicing this to, to get it vetted. And then I think you need to then sample across other environments to test it out.
Well, thanks for that. Appreciate that.
Thank you. Does anybody else have any other questions before we move on? Mark, one more, and then I think we should wrap it up.
No, I don't have any more.
Oh no, Mark Rockwell. Sorry. Different
Sorry.
Mark. Sorry, Mark Capelli.
Right.
Oh, hold on, Mark Rockwell, you are muted.
Brooke, I was just curious when you mentioned that you can't tell the difference between the anadromous form of steelhead or the resident form. Is that due to a specific type of DNA that must be identified with each of them, or because I just heard about a research project and I can't quote any of the details of it that identified gene alleles in steelhead where there was a resident allele and an anadromous allele present that could determine whether the fish was actually going to become a resident rainbow or actually go to the ocean. And that some fish can carry both a resident allele and anadromous allele or have two alleles of each type, resident or anadromous. So I'm just curious as if that anadromous resident distinction will be likely to be able to be made in the future with eDNA.
Well, so essentially it has to do with where we're looking for the information. And right now, eDNA comes from the mitochondria. Because it comes from the mitochondria, it comes from the maternal lineage. And so you're sort of locked in with whatever is found within the mitochondria. So there could be more work done to see if things are going to be, could be distinguished at level. As far as I understand, and I don't want to say it's been fully vetted, but it hasn't been possible. Nobody's found it within the mitochondrial DNA information. That it's, as you say, it's kind of at the finer scale. It's, it's in information that's coming from nuclear DNA that is more quickly turning over. And that because of that, I think in order for us to be able to get at it, I think probably we need to fully vet the mitochondria, in genetics labs that would be looking for the differences, and or it's possible it's been totally vetted, or, and I'm not up on that, or to try to identify where in the nuclear DNA it can be done, and then try to figure out how to sample for it in the field, because that's the real issue with trying to get eDNA from nuclear, is the nuclear genetic information breaks down so fast in the environment that when you go to sample for it in your water bottle or in your flow through pack, you just don't have that many pieces of nuclear information left. And so the real issue is kind of like and I think, probably we need a few lab groups focused on this, trying to get at it to be able to get to see if it is more possible.
Thanks for that, Brooke.
Yeah.
Yeah, thank you very much, Brooke. Super informative. Thank you, thank you, thank you. If you'd
Thanks
like
for
to
the
stay,
invitation.
yeah, if you'd like to stay for the whole meeting, you're welcome to. If I don't know if anybody else has any more questions for you, but we do have an open discussion that we invite you to stay for if anybody has more questions. Okay, is this is last call for anybody? All right, let's move on. Thank you again, Brooke. Amazing talk. And let's move on. So we just had a good talk and presentation with the question and answer session from Brooke. Next, Mac, are you on call? You want to give a quick update? Okay, yeah, you want to just give the slightest update for the strategic planning, what our next step is? Yeah.
Thanks. Yeah, this is just a quick heads up. So the strategic planning process that we announced at last fall's symposium continues. We've provided a couple updates on these meetings, and so just wanted to give you a quick status and then a heads up to watch your email later this week. And so there's a small planning committee that's been meeting regularly. It's about 8 of us that are doing that. So thank you to that crew. We'll send an email out to remind you who those people are. But we've been progressing well. The focus here, just as a reminder, is producing the strategic plan for the Southern Steelhead Coalition to cover the years 2028 through 2032. And that's been funded by CDFW to produce this. There's been a lot of work. We've got what I would say a solid working draft produced by the Strategic Planning Committee, and we're now at a phase where we're looking for more input. And so we'll send an email out with more information about where we are, what it is, what it isn't, and give you an opportunity to weigh in, either with questions. There'll be a link to a survey with some specific questions we have that will help, as we're more into that last, right? The last 20% of any big exercise takes about 80% of the effort. So we've got a good directional heading. Now we're looking for input to make sure we can refine that as we march toward a fall draft submission to the funding parties that are helping us put this on. So that's the update. The ask is this. Watch your emails either tomorrow or Friday from Elizabeth. She'll send a deeper status update via email, along with a link to a survey that if you'd like to provide more input or have more insight, click it. It should take you about 10 minutes. If it takes more than 15 minutes, you're probably thinking about it too hard. And then, of course, if you have any questions, you can reach out to any of us that are on that email. I'll identify who those strategic planning committee members are, and feel free to ask as the questions come to you. Okay.
All right. Thanks,
That's
Matt.
the update. Yep.
All right. And again, just keep an eye out for the email. All right. Thank you very much, Mac. All right, let's move on. So this is a time where we are looking for coalition-wide member updates. Looking for updates on applications on implementation project. Did you put an application in? You have any project updates? We love to hear your project updates. That's what the coalition's for. Tell us what you're doing in your area so we know how to help, how to stay informed, what not to do. And this could be from agency groups, government groups, or NGOs. And then if you need any support, quick update from the coalition. We put an application in for the FRGP to continue funding for this this program. We are trying to extend to cover the whole DPS. So the Southern Steelhead Coalition is actively trying to cover entire DPS wide and merge with the South Coast Southern Steelhead Coalition. That's down in San Diego, who has a nice, great group of membership down there too. And as we go through the strategic process, you will find out more about our governance structure from that as we develop it. So if you're wondering, how is this going to work? Stay tuned. We have plenty of plans in place for that. But then again, if you have any questions, please email me. But yes, things are moving forward with the Southern Steelhead Coalition. All right, let's go to the next update. I think Mark was first. Mark, do you have a quick update for us?
I do. Thank you very much, Elizabeth. And I just wanted to let everybody know that the Santa Barbara Fly Fishers Conservation Committee is focused on cleaning out significant barriers on Mission Creek, a stream that flows through the center of Santa Barbara and then up to the mountaintop. And we've identified 6 important barriers that need to be removed so that fish can have access to spawning and rearing habitat in the upper watershed above the botanical garden. So the good news is that 5 of those barriers are being either managed for barrier improvements and changes or are in the process. And we thank the City of Santa Barbara and the City Creeks Division for the work that they're doing on 2 barriers on Mission Creek that is part of their plan that they're now seeking some design improvements and working towards construction, as well as the botanical garden is working to try to find a way forward for the Mission Dam. And we've met with the County of Santa Barbara on 2 debris basins, one on Rattlesnake Creek and one on Mission. So the last one I don't have a solution for at the moment is where Highway 192, also called Foothill Road in Santa Barbara, crosses Mission Creek. There's a pretty significant barrier there, and hopefully we're going to talk with the Kachuma project about that a little bit in the future. So we're focused on Mission Creek and getting that one fully accessible all the way upstream to suitable habitat. So That's
Thank
us.
you. Thank you, Mark. Good update. Russell, go ahead.
Hey everyone, thanks Elizabeth. Yeah, a couple updates for everyone on stuff going around for Caltrout's projects. And I'm planning to do a more thorough update on the Harvey project at the symposium, but Harvey Diversion is in the process of getting the 75% plan check set in place for the updated design. And we currently are building physical models in Vancouver, BC, 2 different physical models to test the assumptions on that project. And then that work should be done before the symposium. So update there. But I will circulate to the group the opportunity for the day that they do the witness testing. So you can potentially tune in and see the testing of the models. But that project is moving forward. And we've had really good results with reducing the change in the technical floodway. And so that has been a positive result. Caltrout submitted a grant for restoration work on Zaca Creek to get final plans for a fish passage barrier in the upper watershed, and then do some conceptual alternative analysis on the barriers that exist in lower Buellton to work on addressing the need for access and habitat restoration in that. That one became even more kind of poignant. Mark Capelli, Laura Reege, and myself attended a technical working group last Friday for the restoration of the San Ynez estuary. The railroad there is coming to its end of service life, and they have to replace it. And in the process of replacing it, they've suggested they may be open to lengthening the opening of that railway, extending the span at that location. And we're taking comment and input on how we introduce more process into the landscape. So not a lot to report there, but truly the San Ynez estuary restoration via the update of that railroad bridge is a once in a generation opportunity to restore even more intact habitat and what's one of the most intact estuaries in the DPS. So I'm trying to stay fully engaged on that, encourage other people to reach out if they want to stay involved and looking to build truly like coordination and coalition support for pushing that forward to gain the most habitat we can. And then the other thing is, we working with the Forest Service presently to submit a grant application to continue the effort to restore Zaca Creek, getting final plans in place, but specifically having those plans be incorporated with a full recreation update of the site as well, to make sure that we're accounting for that need at that location, considering the importance that Rose Valley Creek has for both Southern Steelhead, but also opportunity to update infrastructure there. I'm trying to rack my brain for anything else, but I think that's kind of the things that pinged up the most. So thanks, Elizabeth.
No problem. And nice to see Rose Valley getting more recreation. Rosie, please go ahead.
Thank you so much, Elizabeth. A couple of fun things to report. One is that Topanga Lagoon is submitting 60% design for the Caltrans bridge replacement. So that's a pretty exciting milestone to be getting to. But that also means that we need to start putting together our funding to actually do the construction. And so the reason I'm bringing this up is because most of the transportation bill grants that are available require a 20% non-federal match. So if anyone has any great suggestions or recommendations on how to reach out and put together a really solid strategic plan for funding that non-federal portion, I'm all ears because we're working on that right now. And, we're going to hit up the usual suspects, FRGP and all of that. But if you have any other suggestions or ideas, please share. And then I see that Isaac Yelchin is on, and we're going to be working, he's going to be one of the leads on doing some eDNA sampling in Topanga Creek and Malibu Creek. We're looking for crayfish specifically. And so, Isaac, if you want to take a quick minute and add something there.
Yeah, we're going to be doing our eDNA sampling in Topanga and Malibu as well, as part of a creation of a larger invasive species aquatic management plan for the two watersheds. And post-fire, there was a big reduction in crayfish, particularly in Topanga. The really high sediment flows seemed to clean them out a bit. And we had some returning of newts as well as spawning of the trout that managed to survive the sediment flows. And so there's a really nice opportunity right now, since there's not as many crayfish as there's been before. So we're going in next week to do eDNA sampling throughout the watershed to try to find where the last remnant populations of crayfish are in Topanga, so we can really see if we can make a big kind of jump on that while they're down right now. Um, yeah, and then we'll be doing similar thing in Malibu as well, but the crayfish are back in Malibu, unfortunately,
Uh,
already.
what was the upper limit? Like how much crayfish did you guys have before this? Was it, do you have any numbers? Like estimates?
We actually do estimates when we're doing our snorkel
Okay.
surveys. I can just tell you they were all over the place and there were lots of them.
Oh no.
But one other thing is that we will be presenting the pre and post fire habitat comparisons in September between Malibu and Topanga. Fortunately, we had data from before the big fires, both Woolsey, Franklin, and Palisades. And so stay tuned for some really cool information about post-fire recovery of habitat.
Yeah. Rosie will be at the symposium. And before I get to you, Russell, real quick, Ken Owen from Channel Islands Restoration said at Channel Islands Restoration, we often we have made matches on grants with the use of volunteers. Ken, can you elaborate a little bit on that for Rosie and for the coalition? Either through the chat or through your speaker if you'd like.
Well, it's just a common technique to do. We had a lot of federal grants, NIFWF grants, and things like that that have had 50% matches. And you can, if there is a role for volunteers, you can count that as an in-kind contribution, and that can count toward a match.
Yeah, thanks so much, Ken. Yes, we've got that one on our radar. Unfortunately, we're looking at somewhere in the vicinity of $80 million of match needed. So while volunteers can certainly help with that, we're going to have to go for bigger and better as well. But appreciate that thought. Thank you.
Oh, that's a good thing. Rosie, please put your email in chat so if anybody has any good ideas, they can contact you. And it's good to get to know the number because I think that will be very helpful with that. Russell, go ahead.
Yeah, that was going to be my follow-up for Rosie with the order of magnitude we're looking at there. I appreciate you giving us information. This one probably is a bit more nuanced, and I encourage anyone to reach out to me if they want to chat about this more. But the comment period currently is open for NOAA NIMS request for comments on their decision that a delisting of Piru Creek below Santa Felicia Dam down to the confluence of the Santa Clara River, remove it from critical habitat for southern steelhead. CalTrout submitted comments in the previous round, fully laying out the science behind why that area was labeled as critical habitat, the critical importance it plays for the species. But they still decided that they issued a decision that it may be warranted to delist that area. Certainly, this is a huge impact for southern steelhead, but also begins to set a precedent for considering the process or reconsidering process by which critical habitat is listed in general. So I encourage anyone that wants to maybe talk about this more to think about submitting a comment letter to reach out. We can have a little bit more of a fuller discussion. There's probably not time here and the ability to kind of get down into the weeds of it. But please reach out if you want to talk about that. Comment letter is critical to trying to protect that section of critical habitat.
And Russell, I think we should add to that the ESA, change of definition of harm. And that's something that all of us are going to bear the repercussions of. So you should definitely be reaching out to your elected officials to tell them about how you are so unhappy that they've changed that definition of harm. And I think the comment period on that is like another week. So sooner rather than later on that one.
Awesome.
Yeah.
Thanks, Rosie.
Thanks for the reminder, Russell.
No, good reminder for both of you. Thank you for reminding about that, Rosie. So yeah, please get out there. There are sites that do that. RJ, in our final moments, go ahead.
Hey everyone, just wanted to give a quick update on Ringe Dam. So a lot is happening with Ringe right now. We are really close to 65% design plans for that, for the dam, and then the upstream barriers component, which is 8 smaller barriers, road crossings, etc., is a little bit on a quicker track. We're just starting permitting conversations and pre-application meetings with all the agencies. So permitting should be kicking off and a lot happening with that pretty soon. We are submitting a grant application for most, but not all of the upstream barriers through the Federal Highways Culvert AOP program, which I'm sure most of you know about. Actually, I think the application is due tomorrow. It's $800 million. They are, divvying out. Hopefully they divvy all of it out, but we'll see. So yeah, we're doing that application, and a lot happening right now, a lot happening in the next year or so. So I think I'll probably give a little more detailed update at our September in-person meeting, but lot happening, and we're making really good, steady progress on that project, and hopefully continue to do that.
Yeah. Rosie, do you have a follow-up for that real quick?
No, but
Yeah.
I just remembered that we are going to be releasing 2 job announcements. We will be hiring
Okay.
someone to help with the Topanga Lagoon project permitting, and we will also be hiring someone to help with all of our aquatic projects. So I will be sending you, Elizabeth, those job announcements. But just a heads up, folks, if you know anybody in the area who's looking for a job, these will be coming out shortly.
Thank you. Actually, I do. RJ, do you have anything else? Anything else about that? Thank you, Rosie, for that. That's really important to know with jobs. They're slim. They're slim pickings right now.
That was it for me.
All right. Very cool. Please, please, please sign up to speak at the Southern Steelhead Coalition, and sign up for registration. That is probably even more important. We need to know how many people want tacos. And that will be TNC's job. So give us a better accurate head count. Please sign up to be a speaker. We're looking to make this bigger and better than last year, and we have some really interesting parties from Berkeley that are going to try to come out all the way down to speak. So please, please, please sign up. Thank you guys for coming. Have a wonderful week. Stay cool. Go to the beach. Get in the rivers. Look for those fish. Anything to enjoy to get out of the heat. All right. Thanks, everybody. Have a wonderful meeting. Have a wonderful day. Thanks for making this wonderful meeting. Bye.